Attention future AP Bio students. We are Christy, Crystal, and Karishma. Welcome to Unit 5 Gene Expression, and we are K-Cube the Science Crew.
Hello, welcome class, welcome to AP Lit.
K-Cube the Science Crew.
Excuse me? Class? Hello, good morning. We have an essay due next week, and you have to check Google Classroom for your assignment. Stop moaning and groaning, come on. I'm gonna do my essay.
Girl, you cray.
What is she doing?
Topoisomerase is an enzyme that controls the winding of DNA during replication. Helicase is a molecule that creates the replication fork. It breaks weak hydrogen bonds, and it also binds proteins to make sure nucleotides do not rebind. The leading strand runs in the 3' to 5' direction. DNA polymerase copies the template that is exposed on the leading strand.
I'm done.
Really already?
Yeah. Are you gonna do your essay?
No. We have so much time.
It's due tomorrow.
Nah, I'm still good.
Primase is an enzyme that adds RNA nucleotide as a primer for DNA polymerase. In the lagging strand, DNA polymerase latches onto RNA primers and copies the template strand. DNA ligase fills in gaps between the Okazaki fragments, which are short, newly synthesized DNA fragments formed on the lagging strand during DNA replication.
Stop lagging. I don't know what to write about.
Here you go. Have your essay. Oh, alright, thanks. If anyone else has their essay, they can hand it in to me.
Can you sign this permission slip?
I said essay. No. Anybody else has their essay?
Here's my essay.
Where were you last period?
I overslept.
You gotta stop lagging.
Alright, teacher.
Wow, this is terrible. This needs so much editing. Okay. Hey, what are you doing? Oh, I'm just grading my AP Lit students' paper. Yeah, while I was grading, I just wanted to call you since you are an AP Bio teacher. I never realized how close writing is to bio. Like, when the children open Word, it's like the AUG start codon. How crazy is that? And then the 4 base pairs are like the 26 letters in the English language. And the 20 amino acids are like the millions of words in the English language. Yeah. I know. And then the homophones are like the amino acids that code for the same protein. Yeah. And then the final essay is the actual protein. How funny is that? And then once the kids print it, it's like the stop codon, 1 of 3 stop codons. Yeah, I just came up with this. It was like an epiphany for me. Yeah.
In transcription, DNA is copied into RNA by RNA polymerase. A template strand is a mirror image of the first, and is where RNA polymerase attaches. The non-template strand is not used as a template, but is identical in sequence to the mRNA, except that all the U's are T's. Genes must be unpacked from the chromatin to be expressed to RNA polymerase. In mRNA processing, the nucleus modifies pre-mRNA before genetic messages are sent to the cytoplasm. There is a cap at the 5' end and a poly A tail at the 3' end. RNA splicing removes introns, which are non-coding regions, and joins exons, which are translated into amino acids. When mRNA comes into contact with the ribosome, translation occurs. Protein is then used within the cell or exported.
Hi, Krishna.
So I read your essay and it seems pretty good. I liked your main thoughts and ideas. There were a couple of grammatical errors and whatnot, but I'm pretty sure you can fix it. It looks good. Just read it over. Let me know if you have any questions.
Okay.
I'll give you till next week.
No, I'll have it done this week.
Okay, that's better for me. All right.
Bye.
Good job. Bye.
Thank you.
Come in.
Hey.
Hey, Crystal. I called you in my office because I have been grading the essays. When did you write this?
Do you want the truth or do you want to lie?
Just tell me, was it last night or the night before?
Try a few hours.
Oh my god. Alright, Crystal. Well, if I'm being honest with you, I was reading this and I don't even think it's English. You really need to translate this back to English, okay? I don't know what language this is, okay? So just look at my comments and edit it, please.
When do you want this by?
I'll give it to you till Friday. It needs to be in, okay?
Okay.
Alright.
Have a nice day.
You too.
Wow. I actually wrote this in a different language. It's in Spanish. How did I do that? Like, really? Was I really that tired? I had to translate this whole thing.
Translation is the process by which RNA strands are made into protein. In translation, tRNAs bond to the amino acid specified by their anticodon. The opposite side of each tRNA, the anticodon, bonds to the matching codon on the mRNA, creating a string of amino acids in the proper sequence. The ribosome makes covalent peptide bonds between the amino acids. The result is a protein chain with the specified sequence of amino acids. During initiation, the small subunit attaches to mRNA at the start codon. The large subunit sandwiches it. Within the ribosome, there is a P site where new protein will emerge, and the A site where amino acid is delivered. Hi, I have an essay. Oh, you handed it in early. Okay. Alright, great. I'll read it.
Okay.
Bye.
Have a good day.
Hi, yeah, I'm leaving right now, so I'll meet up with you in 5 minutes. School's over. Alright. Okay. Oh, hold on. I have a student. I'll call you later.
Here's my essay.
Crystal, what time is it?
It's the bell just rang, so it's after school. But it's Friday.
But it's
Friday.
after school.
You said Friday. You didn't tell me when.
Crystal, I don't stay after school. If you would have came 2 minutes later.
But I didn't come 2 minutes later. I ran for my last period.
Is this one better than the one you gave me before? Is it in English?
Yes, I made sure this time.
Alright, fine. You gotta stop lagging though. I told you that.
I can't help it. It's part of me. I always do it.
Alright, Crystal. I'll read it. Have a nice weekend.
You too.
The lag operon is the inducible operon, usually off to save energy when lactose is not present. The TRP operon is the repressible operon, usually on. It turns off when tryptophan is present.
In a cell, nucleic acids are represented by two separate yet equally important forms: the DNA that stores information in the nucleus, and RNA that is used to translate that information into proteins. These are their stories.
Good day, ladies and gentlemen of the jury. My name is Miss Crimson, and I'm here to prove to you without a shadow of a doubt that my client, Colonel Custard, is not guilty of the most heinous crime of murdering poor Mr. Bones. The host of the International Cookbook Writers Convention was cruelly murdered with a lead pipe in a spiral staircase while on his way to deliver his award-winning cookbook to the kitchen. However, the defendant, Colonel Custard, was not responsible. The testimony of my expert witness will not only clear my client of all wrongdoing, but will also reveal the identity of the true killer of our poor departed Mr. Bones. Your Honor, I call to the stand Professor Pear, an expert in DNA structure and function. Professor, could you please explain to us what DNA is?
Why, yes. I'd be happy to tell you about DNA. DNA stands for deoxyribonucleic acid, and it's a fascinating molecule. It's one of two basic types of nucleic acids, the other being RNA, or ribonucleic acid. Nucleic acids are the molecules that cells use to store, transfer, and express genetic information.
That's very nice, Professor, but could you elaborate on the DNA molecule?
Yes, of course. It's the molecule which stores genetic information in an organism. It's essentially providing directions, like a recipe, if you will, for pretty much everything that makes us, well, us. If proteins are like the building blocks for structures, enzymes, and other cool things in a cell, DNA is like a recipe that tells a cell how to create those building blocks.
So you're saying that everyone has this DNA stuff inside his or her body?
Oh my, yes. DNA is an integral part of every organism, just like other types of organic molecules found in our body, like carbohydrates, lipids, and protein. But the structure of DNA is very distinct from those other types of organic molecules. Deoxyribonucleic acid gets its name from the fact that DNA possesses a sugar called deoxyribose. Now, that sounds like a fancy name, but really it's very easy to remember that it's a sugar like other sugars you may have encountered, such as sucrose and fructose. Just remember that all sugars end in OSE. A monomer of DNA is called a nucleotide.
Professor, can you please restate that definition in simpler terms?
Oh, I'm sorry. A monomer is just the most basic subunit of a complex molecule. That makes the nucleotide the most basic subunit of DNA, or more generally, of any nucleic acid. A nucleotide is composed of three.
Hey guys, thanks for watching Gene Expression. Next up is Juliet, Rami, and Chris with Cell Cycle.